Proteins necessary for gene expression need to access DNA in order to perform their specific functions. But DNA in eukaryotes is not freely accessible to these proteins due to it forming chromatin. Chromatin is made of DNA and nuclear proteins. DNA is wrapped around histones forming nucleosomes, which allows DNA to be compacted in an ordered manner into chromosomes. Chromosomes are found within the nuclei of cells in animals, plants, and fungi. Chromatin is a dynamic structure and its compactness changes over time. Histone modification enzymes and ATP-dependent chromatin remodeling complexes can modify histone proteins and move nucleosomes around, allowing the chromatin to loosen and become more accessible or vice versa. Once the chromatin is open, DNA regulatory elements, such as that in gene promoters, become exposed, so proteins can access the DNA material within the chromatin to perform important functions such as transcription, DNA replication, or repair. DNase-Seq is a molecular technique that identifies accessible regulatory regions on the genome by utilizing traditional DNase-1 footprinting and next-generation sequencing. This method uses non-specific endonuclease DNase-1 enzyme to cut regions of the genome that are exposed and thus sensitive to cleavage. Let's break down the process of DNase-Seq. First, cells are isolated and collected from the target organism. The cell's membranes are broken down by lysing the cells to release the nuclei, which contain the chromatin. Once the nuclei have been isolated, DNase-1 is added in a controlled, low concentration to prevent excessive digestion. The enzyme will preferentially cut DNA in open or accessible regions of chromatin where nucleosomes are not present. After partial digestion, the proteins are removed using phenylchloroform or similar technique, and cleaved DNA fragments are purified. The extracted DNA is separated on a gel, and 50 to 100 base pair long DNA fragments are excised and purified from the gel. Adapters, which are short, double-stranded DNA of known sequences, are ligated to both ends of the fragments to prepare them for sequencing. The fragments are amplified by PCR to create a DNA library, which is then sequenced using Illumina. Illumina sequencing will generate millions of short DNA reads corresponding to the cut sites. The reads are aligned to a reference genome to pinpoint the exact locations of DNase-1 hypersensitive sites. Mapped reads will look like in this graph, where exposed regions of chromatin will have the most reads and form peaks. By examining the peaks within a DNase-Seq graph, researchers can better understand the chromatin structure and identify accessible gene regulatory elements, such as promoters or enhancers. Scientists can identify known or novel transcription factor binding motifs within the open chromatin regions to infer transcription factors that regulate specific genes. Scientists can compare data from different cell types, conditions, or time points to understand how chromatin accessibility changes, and they can integrate DNase-Seq data with other data sets such as ATAC-seq or RNA-seq to build a more comprehensive understanding of gene regulation.